So I wanted to start just by pointing out that there's a lot of different types of transcription factors, and they utilize many different strategies when interacting with DNA. So I'm going to test my aim from over here with this pointer, but, oh, I don't have to. Okay, so there are, here I'm showing you different families interacting with DNA. These are all based on crystal structures. The bZIPs, for example, interact with DNA in this classic kind of zipper type grid. There's AT hooks that only use a few amino acid residues to interact with DNA. And then things like STAT that have much more complicated modes. So just a wide variety of families that have all kind of evolved very different strategies for recognizing DNA sequences. And then likewise, if you look across the eukarya, there's a very wide distribution of these families across the organisms. So over here is a phylogenetic tree just giving representative organisms across the eukaryotes. So there's fungi up top, animals, plants, and protist type things down here. And now what I'm showing you here is there's about 80 or 90 different families of transcription factors. And each entry here tells you how many of the given class of transcription factors are present in the given organism. And the scale's over here. And just kind of again, so blue means that the family's not present at all in that organism. And the take-home messages from here is there's kind of like this kind of class of ancestral transcription factors that seem to be common to all eukaryotes. And so maybe they recognize the original toolkit of transcription factors. But then there's lots that are specific to different classes. So there's animal-specific transcription factor families, plant-specific, et cetera. And kind of the important thing for this talk is that for the vast majority of these, we have no idea what DNA sequences any of them can recognize. So I did my postdoc in Tim Hughes' lab, and the assay of choice that we use for determining transcription factor binding specificities is the protein binding microarray. So just in brief, the basic idea is you make a plasmid that usually contains the DNA binding domain itself and then the 50 amino acids flanking it. And then you use this to bind to a DNA microarray, and then based on which probes light up, it tells you something about the specificity of the transcription factor. The way the probes of the arrays are designed, there's about 40,000 different probes. Each has about 35 unique DNA bases. And the way they're set up is that every probe, when you look across the probes, they contain all possible 10 base sequences. And therefore, every possible 8 base sequence shows up 32 times in diverse sequence contexts. So that way, it's a nice kind of statistically robust way of sampling the entire space of possible sequences for a given transcription factor, what they like to bind to. And there's lots of aspects that we like about these where they're reproducible, and then the scores track fairly well with the binding strength. These were originally developed in Martha Bullock's lab, but when I was first joining Tim's lab, we were working on ways to kind of ramp up the throughput of these. So, and this is about when I was joining Tim's lab again. This observation was made by Tim and Martha that it's an intuitive observation, but an important one, which is that if you have a similar DNA binding domain, you're going to recognize a similar binding motif. So this is illustrated quite nicely here, where they did basically all the mouse homeodomain transcription factors. They ran about 160 different protein binding microarray experiments to figure out their different motifs. And now this tree here is just showing you what you get just by clustering the amino acid sequences that are DNA binding domains. And now I'm superimposing the motifs that they recognize, and they're color-coded, so you might not be able to see them all, but the idea here is that if you have a similar amino acid sequence in your DNA binding domain, you're going to recognize a similar motif. And that's true as you march all the way through all the 160. With very rare exceptions, there seem to be these pretty strong rules where if you cluster together based on your protein sequence, you're going to recognize essentially an identical motif. So that led to this idea of, it was called at the time the 65% rule, which means if you take a pair of proteins and align the amino acid sequences in their DNA binding domains, if it's greater than 65% identity, then you're going to have a nearly indistinguishable motif. And there's some really interesting aspects of this. So for example, it has nothing to do with evolutionary distance. So for example, there are plenty of examples of yeast and mouse homologs with identical motifs they recognize just because there's been strong enough constraint on the binding domain that it hasn't changed over time. This paper down here, Elaine et al. Evaluated several different ways to try and improve on this simple alignment method, but the punchline from that was that the alignment method, you can do slightly better with more advanced machine learning methods, but the simple rules perform pretty well, which is nice because, as you'll see, we can ramp this up on a much higher scale and run these quicker methods. If you saw Tim's talk on Friday, this kind of thing, we've shown now that it works well for RNA binding proteins too, and this paper was just recently published. So what's nice about this is, if provided that you knew this rule, you can imagine doing a broad survey of the different families of transcription factors. So that's exactly what we set out to do. So this is again the eukaryotic tree with fungi up top, animals and plants and protists down here. And what I'm showing you is if there, at the time when I made this slide, and there's probably more now, but if there's at least one genome available on that branch, then there's a check mark. And then the stars indicate collaborators that we were able to chase down that could send us RNA or cDNA or some sort of template that we could use to create a construct to run a PBM for a protein from that organism. So you can see pretty much anywhere where there's a sequenced genome, we were able to find someone or multiple people that could send us something to run our assays on. And this is just a pretty picture to show you the wide variety of organisms that we're including. So, we're looking at pretty much all forms of eukaryotic life. So when we set out, we decided, well, how are we going to choose from all the transcription factors? About 160,000 transcription factors when you add up across the 300 or so sequenced eukaryotic organisms. So, we're not going to be able to figure them all out. So how do we choose them? So what we did was, so the first thing we wanted to do is figure out exactly what these inference thresholds are for each family. So I pointed out that for homeodomains, it looks like it's 65%, but maybe it's different for different families. So you can imagine kind of choosing different pairs at different thresholds of amino acid similarity and then assaying those. Second is, we want to kind of maximize the bang for our buck here. So, if you choose a large family of transcription factors that there's no motif, then we'd rather do that than targeting ones we do have a motif for, or smaller families perhaps. Third, we wanted to kind of do a good job of sampling across clades, so we didn't want to only focus on specific branches, although we did focus on certain model organisms just because we thought those would be the most useful. And these also happen to be different organisms that we had the most enthusiastic collaborators for. And here's the results, just again broken down by the different organisms here, and these are all different families. And the entries here are just showing you how many transcription factors we got a motif for the given combination. And you probably can't read this, and that's okay, but I just wanted to point out the species are color-coded, so different colors indicate different kind of branches of eukaryotes. You can see, we're not only doing metazoans, we're not only doing plants, etc. The total number that we got, so no assay is perfect, so we targeted about 2,000, and we ended up getting about 1,033 different transcription factors. And again, these are done in duplicate on our 2 different array designs. These cover in total about 130 different species and over half of the transcription factor families. So let's take a minute now and revisit the 65% rule. So again, the idea here is this axis is showing you how similar the amino acids are in your DNA binding domain, which again is just the percent ID in the alignments. And in this axis, we've tried several different ways of measuring this, but it's basically how similar is the DNA motif that you recognize. And in this case, I'm going to show you what percentage of your strongly bound 8-mers are identical. So what I'm going to show you here, so for homeodomains, I'm showing you 1 bin at a time. So looking just as 100 bin, these stars are going to indicate the precision, which is essentially what fraction of the protein pairs in that bin are as good or better than most experimental replicates. So if you do replicates of the same transcription factor, just because of variability in the experiments, you might get slightly different answers. But this is a pretty stringent way of saying you have to be as good or better than the majority of experimental replicates for the same transcription factor. And then these are standard box plots, so that'll show you the median for the bin. And then down here is just the number of pairs. So for example, here we had 86 pairs of homeodomains with identical DNA binding domains. And this is how it kind of dials out. So for homeodomains, again, this dashed line here, we said, well, let's require 75% precision in a bin. And you can kind of march along then and see where the precision drops below that. So right here, and then you can look down and say, okay, well, anything above 70% amino acid identity, we can say you're probably going to have a nearly identical motif. And then you do indeed get very different answers for different families. So here for MIB, we kind of got unlucky here where this one just falls right under the threshold, but we're fair, so we say, all right, well, the line's right there. Maybe if we do more experiments, we can push the line out a little further. For bZIPs, you can go a lot further. And for all the different families we looked at, there's different cutoffs, but the point is that these all kind of have this kind of sigmoidal type shape, so that the rule seems to hold. There are some interesting examples, but in general, we could figure out rules for the vast majority of different families. And now, since this is a systems biology meeting, I got to show some networks. But this is just, I think it's an intuitive way of presenting this. So we're just looking at two different subfamilies of bZIP transcription factors. Each node here is a transcription factor, and then there's a line between a pair of transcription factors if they're above that threshold of similarity that's required. So you can see here we have a big cluster of bZIPs from the June D family, and I'm just superimposing the motifs we get, so not surprisingly, they're all highly related, and the motifs, as predicted, are nearly identical. And then these are, again, these are also bZIPs over here, but it's a different subfamily of bZIPs, and as predicted, there's no lines between the two groups, so they have a different looking motif. So zooming out a bit, so we came from over here, now we're looking at all the bZIPs across all eukaryotes. The colors of the nodes tell you the status of that transcription factor's motif. So a red node is something we've directly run an assay for, or someone else like UC with CLEX. We've compiled pretty much all the motifs we can find. Orange are ones that are linked to a red node, so it means we think we can infer a motif for them. And then blue, dark blue, are new proteins from families that we targeted for this study and got a motif for. And light blue then are ones that we can now infer based on our new data. So you can even see kind of at this resolution, we're doing a really good job with the bZIPs. Most of these families, the larger ones at least, we have at least one motif for. And then you can zoom all the way out, so the bZIPs are up here, but these are all the transcription factors now. And again, even at this resolution, you can kind of, if you squint your eyes, you can see there's a lot of red, orange, and blue in here, meaning that we're covering most of the major families of transcription factors with motifs. And here is exactly the coverage that we have now. So this axis is just going from 0 to 100% coverage. We're sorting here by the number of proteins in each family that we targeted. And the different colors again, so blue is previously known, so you're not going to see a lot of blue. This is again compiling everything from Transfac, JASPAR, every publicly available study we get our hands on. Red are new transcription factor motifs that we've determined, and then green are ones that we can infer from either the red or the blue ones. And it's, you can see that there's lots of inferences we can make because transcription factors in general, there's a lot of duplications where the families will maintain their binding domains. So in plants especially, there'll be large subfamilies of transcription factors with hundreds of members, even just in a given organism. And then also, like with primates, there's not a lot of evolution going on. So if you know a human's transcription factor binding motif, you can often map it to chimp, gorilla, et cetera. But you can see, for the different families, we're doing pretty good. This is a function of course, how well characterized the family was to begin with, and also what the cutoff is. So families with more stringent cutoffs, we can't make as many inferences. But we're also doing pretty good on our coverage by species. So again, this is the eukaryotic tree. We have fungi up here, animals, plants, and protists. And you can see, like for Cerevisiae, of course, there was a lot of information already known, so that's why there's a large blue bar. But for the majority of these branches, even down for the protists, we're starting to get some useful coverage of transcription factor motifs. And this is kind of just putting it all together just for the final numbers. So if you look across, there's about 300 sequence eukaryotes when I started this project. There's about 162,000 different transcription factors in these genomes. And if you take everything that's available in the literature, you maybe have about 2,000 or so, which is less than 2% of them with a known motif. In our study now, we have either determined or can infer we're up to about 63,000 transcription factors with motifs, which is almost 40% coverage of all eukaryotes. As I mentioned, we kind of have a stringent cutoff for these inferences. If you lower this down to 50% precision threshold, we have coverage of almost half of all the transcription factors. This is an ongoing endeavor where we've recently just continuously trying to target large families and subfamilies we don't have information for, continuously trying to update these inference cutoffs, so we have some more proteins on the way. And we think this will be useful for people, so we spent a lot of time making a website. It's still not quite published yet, but if you're interested, you can talk to me afterwards. But you can browse based on your favorite transcription factor or organism. It'll take you to every transcription factor has its own page showing the motifs. And we have bulk downloads, so you can easily download everything. And we have different tools for scanning DNA sequences or predicting motifs based on amino acid sequence, et cetera. And I'm going to skip the EQTL thing because you guys heard a lot about that already. But we can use these to try and explain EQTLs. Okay, so thank you for your attention. This has been a large endeavor, so I divided this into University of Toronto and not University of Toronto. But this is everyone, lots of people in Tim's lab that have helped with this. Tim, of course, was instrumental in this, and Ali Yang did most of the experiments, and Mihai helped a lot with developing the website. And then these are all collaborators. And if you want advanced access, again, just contact me. And I've recently started my own lab at the University of Cincinnati and Cincinnati Children's Hospital. So if you're interested in doing a postdoc, just contact me, please. And that's all. Thanks.
Let's thank the speaker. I think the question will have to be very fast, and maybe we can do the rest on the break because we have.
All right, very fast.
Well,
it did take 17 minutes.
Yeah. Okay.
Sorry.
Okay, I want to say
I had 20 minutes though.
I've been one of the lucky people to get early access to this database, and it's really amazing.
All right, thanks.
One question I had is, have you done any of the transcription factors that have been done by Jaspar and found, and if so, what was your agreement between your motifs and their motifs?
Yeah, so we were extra careful to make sure there's a good overlap with what the answers we get and what they get.
Great.
And I had 20 minutes, by the way, for my slot. So,
oh,
so Manolis.
I'm sorry. So if there are more questions, then sorry.